but that won't stop me for later. So tonight will be more of getting back into that grind and be more focused on that this week
Hmm.
after the little break. So
Yeah. What does your like holiday rest of December look like? And like how productive, like I don't know if you have travel plans or like family plans or anything, like how productive do you think you're gonna be kind of in the next 3 weeks or so?
very productive considering we don't really do
Oh,
anything. I think only Christmas Eve is the only thing I have
okay. That's
something
good.
going on. Everything else is just based around work, and that's the same Tuesdays, Thursdays, unless there's something canceled or like postponed because of holiday stuff. But no, Christmas Eve is
Gotcha.
the only one that's in the way. Other than that, solid.
That's honestly good. I was studying, I took mine like in January. Christmas break like killed my momentum.
I
Because I
bet.
was like going home from college and everything and like not in my normal study space and everything. So
It
that's
switched
good that you're at
everything.
least gonna have like some continuity of life.
Yeah.
Um, so okay, that's good. Um, yeah, and hopefully you can find like fun stuff to do too. Take nice little
Yeah.
study breaks.
Not too worried about it. If anything, it will give me more of an opportunity to even like find more studying spaces considering everybody will be gone. So I could crash the underground campus and study. And if we're doing practice exams, I could easily also um have that as an opportunity for another place. Because I think I learned from this diagnostic is that I need to not do practice tests from home anymore. I don't think it's going to be any benefit, honestly. So secluding
Hmm.
myself and also interpreting those breaks, not like I haven't done the breaks before, but I have, but also just, I mean, starting early helps with that momentum.
Yeah, and I think I like you think about, in psych, there's that concept of like context dependent recall. Um, I was a big fan of like not studying ever in the same place, like kind of
Agree.
bouncing around a lot.
Um,
Um, so I would just like, I probably studied in every seat in my library at some point, like just bounce around everywhere.
Bounce around. Yeah,
Um,
that's smart.
yeah. The hard part though is just finding like an uninterrupted public space for
Yeah,
like 8 hours. So you do
precisely.
what you can.
Yeah. And I don't know,
Okay.
worst comes to worst if they're going to get mad about it. I'm an alum at the university here in Buffalo and also in Rochester. So I don't see why they would kick out the alum. So I could just show them my past ID and be like, I just need a space to do my own thing. And because they're weird about library hours here, like public libraries. They they open late, like around 10 or 11, and they close early, like 4 or 5.
That's so sad.
I know. So I'm like, I either have to go home to a cafe or I have to go roam around a university campus, and it sucks, but
Hmm.
we'll make it work. It's fine. I'm not worried about it.
Yeah. Yeah. It'll be nice if there's not like a ton of undergrads too, if everyone goes home.
Yeah, that's kind of the reason why I'm like not going back because it's different. It's a different vibe. But then also with the graduate place, the schools they have limited studying spaces as well, which is sad. So
Hmm. Okay.
take what you can.
Well, yeah. Good luck in your searches.
Yeah.
Okay. Cool. So we can think about like breaking down the like remaining practice tests then I guess semi soonish.
Mm. That's
I
totally
don't
fine.
think um yeah, because I'm looking at the like syllabus we have, which is not necessarily super strict, but
Also not updated.
yeah.
But like I do want to keep that where was it? That one spreadsheet that I told you about. I know you looked at it at one point and I don't think I have it open at the moment, but that's what I'm gonna
The FC
do
reviews, the foundational
basically.
concept review.
Yeah. So I want to keep that because I think that will help with the gaps of bio and biochem because it's really biochem focused and also consistent with all the other topics. And if I need to, I'll adapt and I'll keep you updated on that. But I don't think I've given enough time for it to actually say if it's working or not, considering it's been maybe 5 days that I gave it a shot. So I'll
Yeah.
keep up with that as of this week and see how that
Okay.
goes.
Yeah. Did you get through all the physics videos and stuff?
Yes.
Cool. Awesome.
The
Okay.
physics
Yeah.
actually,
Yay.
I know. I'm like, now I can finally actually practice and not worry about this one extra little thing to do on my list. So
Mhm.
that will be very much relieving, honestly.
Okay. So then I might just type this in the foundational concept review one because I feel like that's a better format. Just thinking about the full length exams, I kind of want to map that out.
Mhm.
Okay, so you've done the unscored, which means you have scored and
Mhm.
then 1 through 4.
Mhm. And I think there's only two March dates. It's like the 9th and another one, not the week following, but like the week after that.
Mm-hmm.
So
I'm pulling it up right now. 2024. Ooh. Yeah,
gross.
March 9th and March 22nd.
Yeah, that sounds right.
Okay.
I
Is there one
haven't,
that you're leaning towards
yeah,
or
I was gonna say I haven't registered towards any for anything. I mean, sooner the better. I'm just more worried about the burnout if I push it to the 22nd, but that's only 2 weeks of a difference. So I don't know.
um I think low key if you do the 22nd, you can space out your practice exams a little more and that might help with the burnout rather than like I mean, I know it's only 2 weeks, but that would give you like 2 less weeks of like straight practice exams.
Right.
Um, but also, yeah, that's just kind of personal preference. I also don't know if you care if it's like Friday versus Saturday.
Yeah, I'm just more because I'm used to doing the practice test on Saturdays. So that's why, and then all the other full exams that I took that were actually through AAMC were Saturdays. So I don't know if it's going to throw me off that I do it on a Friday. That's the only thing because I'm just really weird about consistency like that.
That's really fair. Okay. I like the 9th then.
Is that the
Not too
Saturday?
like, yeah,
Okay.
it's not too like far out. I mean, it's far enough away that you still have time and you don't have to panic. But not too far out that it's going to be like, gosh, I can't make it this far.
Yeah. Okay. That's totally fine. I didn't register for either one because I wasn't sure what the official target date would have been. But since we were more leaning towards March anyway, I was kind of in my head ballparking March 9th anyway. So,
Mhm. Yeah, and I guess like worst comes to worst if something goes really wrong. There's like 4 April test dates and 5 May test dates, which
that
is
was feasible.
nice. Like I don't think we'll need those, but just in case.
Yeah, as a backup
Always,
plan. Sure.
yeah.
Also, I was thinking my birthday is April 22nd. So I'm just thinking, like, what kind of a present would that be if it didn't work out?
Yeah. I took my exam January 14th. I turned 21 2 weeks later. So I had a great time
That's
because
amazing.
I was like done with my exam and then I could legally go out. And so it was just like so much fun.
Oh my God. That's perfect.
And then I got my scores like 2 weeks later. So it was like I didn't even have to worry about the scores yet. I could just like party.
Whatever. Yeah.
But anyways.
You were still in break during
Okay.
that time, I'm assuming.
Um, my classes had started like that week. It was like the
Oh,
Saturday of my first week of classes. Um,
okay.
but it actually kind of worked out because I didn't want to study super hard that week of the test. So I just kind of like went to class and it was all like the syllabus week. Like you know,
Nothing
stuff. And
hard.
so
Yeah.
just kind of chilled. Yeah, it was not too bad.
Good.
Okay, let me pull up a calendar to put side by side this thing.
So then what would you suggest for me if we're aiming for March 9th? Regarding work, I just want to make sure I put in my time, like to request my days off early in advance. So what would you prefer I do regarding when I should be work free while prepping for this? Like, should it be 1 week or 2 weeks?
I think if you take off, because you're just Thursday, Friday, right?
Tuesday, Thursday, but yeah,
Oh, okay. Yeah, Tuesday, Thursday.
you're
Yeah,
totally,
wait, but I knew that. I don't know why I looked at that. Okay.
you're so fine. Don't
Um,
worry about it.
yeah, I think if you just do like that week, so that would be like March 5th and 7th. I think
Okay.
that's manageable. Yeah, I don't know. I feel like I don't want you to like be sitting around with no work and just be like stressed about the exam.
That's valid. Yeah. I'm actually, yeah, that's valid. Yeah. I can't argue with you on that. Nice request the 5th and 5th and 3. I'll do that tomorrow. I'll put that in the schedule.
Mhm.
Okay, cool. So I guess that's
Okay.
officially like 3 months out as of this weekend, more or less. Okay. This weekend.
January. Okay, I'm just putting in months that we're in.
You're totally fine.
January 14th. Yay, and then hopefully you'll be done with all this by your birthday.
Oh my God, that would be the best 26th birthday ever. Oh,
Mhm.
it'll be so great if I could just like put the MCAT to bed and just roll with applications at this point.
Mhm. Honestly, the applications, at least the first part of applications, is like so low stress,
Yeah,
which is nice because it's just like, I don't know, college apps 2.0.
exactly. Yeah, that's what I've heard at least. Yeah,
There's some like stupid prompts, but you just kind of live with it.
yeah.
March. Okay, so this would be test. Cool. That
Oh my
puts
God,
us
that's like
at,
it's 13 weeks. Jeez.
okay, that's a good place to be. And a good, I'm glad we did the diagnostic, like, um,
Earlier, like within the past couple of
yeah.
weeks. I'm just really happy with where that's placed. Like right now, it's a solid 500 and working at least. Like I would love it if I get a 512 at least. Because I think that would be more of a considered, like they say 510, 511 is more of a considered of a competitive applicant, but 12 is my favorite number. So I'm like biased. And I'm like 512. But
I
I don't
feel like it's achievable,
I think so
um,
too,
with
honestly.
bio,
Yeah,
because
I agree.
bio can make up like, I've
Like
always seen like people who have that like 1 week section. If you can like flip that week section, that's already like 5 to 6 points, like just right there. Um,
I agree.
okay. So I think what I'm leaning towards, do you need me to share my screen or are you like following along?
I'm also following on the document.
Okay, cool. Um, is generally doing AAMC 4 probably like the week before or 2 weeks before because that's the like this is or the AMC scored one. That's like this is the most accurate, most recent one.
Mhm.
Um, and so then you get a good like, okay, my test is in a week, like this is where I'm at. Set my expectations realistically from here.
Mhm.
Okay. And I think what I generally am leaning towards for you would
Mhm.
be doing a full length almost every other week
Mhm.
rather than every week.
That's fine.
Um, I think you do would do well. and correct me if I'm wrong, with this more like spaced out, like really having time to dig into the concepts and like go through review and stuff. Um, that's just generally kind of how your learning style strikes me.
Honestly, that works well. I think that's doable easily.
Okay, cool. So that would put your next full length, like the first week of January.
Okay, for around New
Um,
Year's Eve. Cool.
and I think this could give you a little bit of flexibility because you have every other week if you need to like move a test around or something, depending on your like weekend plans.
Mhm.
Um, so you could always like move one forward or backwards a week kind of thing, and still stay pretty on target. But that would be so basically like like technically 3 tests in January, but like early, middle, very end slash start of February, middle February, end of February, start of March is your test.
Yeah.
Okay. So for the AAMC material, you do have to do the like purchase the whatever access, which I'm sure you've done before, and it should last you a year. So I'm pretty sure I don't think they give you any like other options. So
Okay.
whenever you um, are like ready to purchase that, that'll definitely last you through your whole like review cycle.
Okay.
Um, and then that will give you access to the question banks and question packs, which will be good to start digging into like now, I think, or soon.
Thank
Let
you.
me check on your U World.
Where are we at?
Because U World's a good like I think how I ended up doing it, just in terms of like my schedule, was like I still had U World to finish, so I kind of alternated between like U World and AAMC stuff because I didn't want to like burn through all the AAMC material right off the bat.
Yeah, yeah. I was gonna say that was more would be a concern, or not even a concern, honestly.
Okay, I don't know if it actually tells me how many questions you have left, so that might be from your side of things if you can see that.
Again, I'm screwing up my password, so give me one moment.
No
Wow.
worries.
I need to put this on my sticky note and just stick it to my laptop at this point because apparently I have so much trouble with this stupid password. Okay, logging in right now. It's maintenance. Okay, cool. Yeah, I have the Q Bank until March 21st anyways, so that's useful. Unused 2,610.
Okay.
Do you also want to break down amongst like by subject / sections of each one, or
Um,
I could also put
do you
it in.
actually just want to like share your screen? Let me give you the
I can also do that.
privileges. I just need to. Okay, you should be able to now.
Cool.
Okay, it's like a good breakdown. Okay, I don't care as much about the cars, which is fine.
I
But
haven't
yeah,
touched the cars because of Jack Weston.
yeah, I think
But
that's fine to keep up. Um, yeah, I know. Of what some people will do is mini like section practice tests within UWorld. So like clicking biochem and generating 59 questions and just kind of going through it like a practice test.
mhm,
Um, I know at least on your schedule, you definitely have like 30 question chunks, right? Is
yeah,
that kind of where you've been at recently?
yeah.
Okay.
That's how it's like you said 20 minimum, 40 maximum. 30 is like that sweet spot. So I aim for 30.
Okay. Yeah, I mean, I think so if you did 30, you'd have to do 87. Sorry.
You're right.
You heard someone outside.
You're fine.
Okay. Yeah, so 30, how many days is there until March? Days until March. 1 sec.
You're fine.
Okay.
I looked it up
There's
for you.
someone.
Oh, was it somebody? I was just gonna say it was 89 days, but go ahead. What was
I
his
was gonna
intent?
say there's someone like right beside the window talking. And so she was like going crazy.
Oh, he wants to join. Mine's just
All good though.
sleeping. Mine's just like, you need to study. He just gives me the sign and crawls underneath my desk. And that's when he knows I need to study. He's like, you're not studying, you should be. So he goes under my desk.
Cute.
I'm like, you're giving me the guilty conscience. Stop it. Anyway, um, 89 days until the 9th, if that's what the
Mhm.
date we're looking at. I don't know if you were looking at like
Yeah.
the 3rd or not. Okay.
Yeah. Okay. I think for now we'll stick with that like 30, 40 max. Have you been doing these with any kind of time limit or timing or anything?
With science, no. But I have
Okay.
been keeping track on my individual, like on the on the UWorld sheets where I list out like what I did wrong, how I did wrong. I do list out how much time it takes me to go through it. So I haven't
Okay.
done timed, but I have been listing untimed, if that makes sense.
Gotcha. Okay. Yeah. I mean, I think as we kind of get through end of December, starting in towards kind of that full length season,
Mhm.
we can think about, I don't like doing it timed in the UWorld app, but like setting a timer for yourself kind of thing.
Mhm.
Um, and going through from there just to get things a little bit more um, kind of efficient.
Got
Because
it.
I know with the full length, it's just kind of like getting that like a little bit of efficiency going to get those last like 10 questions in or whatever, right?
Absolutely. Yeah.
Hmm. Yeah. And I found if you struggle with timing, one thing that we can actually do kind of going forward is if you do them in like small chunks and then set the time limit. So it's whatever, it's 59 questions and you have like an hour and a half. And so you just kind of like do the math on how long each question gets. I forget exactly what the numbers are. And do
Sure.
like a five question chunk in like, here's your 7 minute time limit kind of thing.
Hmm.
Um, and like go through like that. That's a good way to like build that up without having to sit down for an hour and a half and like
Do it.
exhaust yourself.
Yeah.
Mhm.
Okay. I mean, I can even
Yeah.
do that with the 30, 40. I can break those into like 15 questions. And then I have it written here, actually. Something like 15 questions. So it's like maybe 20 minutes, something like that. Because 10 questions is about 15 minutes and 20 questions is about 30. At least that's what I
Okay.
have written here.
Yeah, I think that sounds about right. Um,
So I could do that. I could even start like how you said to put it like on the side. It'll just probably be broken up into two tests, but it will be 30 or 40 questions.
Hmm. Yeah, so it's at like about a minute and a half per question. So yeah, if you did 15 questions, then, or would you say like 20 questions would
Well,
be
20
like 30
questions is
minutes,
about 30 minutes, and then 15 would be 22,
20,
23.
yeah, 22 and a half.
Yeah.
Yeah, that one you can go for like 23, 24 kind of thing.
Mhm.
Um, yeah, there's also one reason why I don't necessarily like doing it with the UWorld timer is there are intrinsically like some questions that are going to take you longer and some that will take you shorter. So if you can get to the point where you have like, okay, this one was 25, but this one was 20, I'm in a good place kind of thing.
Mhm.
Um, I think that'll be good. And then if even that is feeling too much, then we can try like even smaller like passage chunks.
Okay.
So like one bio passage, here's 10 minutes, go kind of thing.
Got it. Okay.
Okay. So I'll keep you mostly doing what you're doing then for the rest of December in terms of your kind of UWorld foundational concept review.
Mhm.
As long as you feel like that's working well for you. I know you said you're still kind of feeling it out, but um, just kind of using these same study strategies that we've been talking about.
Mhm.
Cool. Okay. Yeah, I mean, I feel like that's at a good place for like the rest of December. I think in terms of our sessions, um, for December, we can try to do some like molecular biology focused, like more content type things.
Mhm.
I know some of the stuff we've done in the past or a lot of the stuff we've done has been very like question oriented. Um, but the way I kind of like to approach like the content side of things is we would like pick like one passage and like go like in over the top detail into like, oh, they just mentioned mitosis. Let's take 30 minutes and talk about what mitosis is, like that kind of thing. Um, so if that would be helpful, I think that could be a good like kind of sit down and explore some of those bio gaps together and talk through um, kind of those concepts and really make sure that those ideas are solid.
Okay. That's completely doable on my end.
Cool. And I should be pretty free um, because I need to escape my family during Christmas break. So,
Valid. Very valid.
okay, cool.
You have other
Well,
clients aiming for March?
um, think so. I like right now I only have one other student. Um, and she was gonna do March, but I think she's pushing it back more because she hasn't started studying yet, like
Oh,
very much.
boy. Okay. Valid.
Yeah. So, but I'm trying to get more students, so we'll see.
Gotcha. Okay. But yeah, I think this is a good plan for December.
Okay, sounds
And
good.
I'll readjust whatever I have up here whenever if I need to. If not, I mean, I'll keep you updated no matter what. So okay, this
Mhm.
is done no matter what. So yeah, I think you'll be fine. Um, yeah.
Cool. Okay, you can hop to the full length then.
Okay. Do you want to share yours or do you want me to continue on mine?
Um, you can share up to you, actually. If you want to type in your little review spreadsheet, we can do that.
I just don't know if there's anything you would want to continue, like to draw, because it's easier for you to do that versus me. That's the only thing,
I was
like
gonna say,
label.
I think I can draw. I think I can draw from my screen on yours, but let me double check that.
Oh, okay.
Let's see.
Okay.
Cool.
All right
Yeah.
then. We're on 28, so we just started. Okay. Is this too small for you? Do you want me to zoom in?
Maybe just a little bit.
Yeah, that's totally fine. No, why are you not? Is that better?
There we go. Yeah. It was like blurry when it was zoomed out. That
Yeah,
works.
no worries.
I don't think I sent you the link to that Jack Weston extension though, but I
Uh,
will. Not that you need it, but it's helpful.
that's true.
Do you have Chrome?
I do.
Cool. I've just found it to be nice to have.
I will copy that link. And I will definitely do that. Very cool. I just don't want to waste time doing that right now, but
Oh no, you're totally good.
I also know that this might not be the best way, considering I don't have another, like the Jack Western, it's just going to be AAMC's description of everything.
No, that's okay. That's what like our review, I guess, is for.
Mhm.
Cool. Okay. This one was correct. Do you just want to glance over it and just feel how confident you were?
I remember having this question or like going through this with my metabolic stuff was that it's better with aerobic versus anaerobic because more ATP would be produced. I knew that. It's all based
Yep.
on ATP. So 28 aerobic versus anaerobic. This question was also right. All right. Okay. Mhm.
Most likely explains why the bacterial colony did not grow immediately after the polysaccharide was added. Okay, so you hit A.
Mhm.
Okay. So I guess just thinking in terms of like population, if bacteria were dying, wouldn't that like dip the population before it then rose?
Oh, I
If
guess
that
you're right.
makes sense.
Yeah.
So if I start with like 10 bacteria
If
and then
they
I dip to 5 and then it would grow, it would be like a whoosh. But instead it's like stagnant and then grows. So I
mm,
think that's why it's indicating that, yeah, it says for A, like there would have been a drop in the curve. Um, as the curve stays horizontal, it is not possible that bacteria have died. So I think what it's getting more at is more of like some kind of temporal restriction. So that's going to be like, oh, stuff takes time to happen.
I understand. Okay.
Does it make sense why, like, yeah, why B would make sense of just like it can still digest it and everything. It just takes time.
Yeah. Because
Mhm.
other, like you said, other things would be happening.
Mhm.
Okay. Because the number of cells are still consistent. Okay. Um, um, more of a brain comprehension slash didn't know.
Yeah. I don't know if you qualify that as like data analysis of like the drop in the curve, but um, didn't know. I think both are fine.
Yeah. Okay. I can go through those later. Okay, cool. 30. That makes sense though. I
Mhm.
should have known that if they were gonna die, though, that means they're gonna be less. Cap DH gene is continuously expressed where is it most likely found? dehydrogenase, well, if it's continuously expressed, it means that gene expression is happening. So where is gene expression happening in euchromatin?
Yeah.
So
basically wants to test your like U versus heterochromatin.
yeah. Sweater tonic at pH 7 despite possessing 2 nitrogen atoms in its formula. So this one's basically knowing, do you know your amino acids based on the abbreviations and their structure / what's the structure at pH 7?
Exactly.
And all of these arginine, lysine are basic. Glutamine is a polar, but that's the one that has 2 nitrogens. Well, I mean, lysine has 3.
Mm-hmm.
No. Yes, lysine has 3.
Or arginine has 3.
Or is that arginine has 3? Lysine has 2.
Mm-hmm.
Yeah. Okay. So it does, except it's not zwitteronic. It's +1.
Yep.
Okay, so we're doing well with amino acids. Or I'll say zwitter. All right, passage.
Okay. We're looking at antigenic variation. Okay, so this is definitely going to be more like molecular kind of transcription, translation, expression kind of stuff.
Mm-hmm.
Okay. This whole intron. Okay, cool. You can scroll to the question if you kind of remember this.
Mm-hmm. Okay. Yeah, I remember going through that. All right, based on the passage, switching of our gene expression probably most directly benefits bacteria by reducing the parasites blank.
Okay, so you said C. So why don't we scroll up to the top, I guess, where it introduces this gene and kind of why it's important.
Yep, saying it's mutually exclusive, gene express consequently the antigenicity of specific PFE-MP1 on the erythrocyte surface, which is throughout an infection.
So I'm looking both up here. Antigenic variation is a survival strategy to avoid destruction by the host immune system. Blah, blah, blah. Surface adhesion molecules. So does it make sense that like if you express different surface adhesion molecules, you're like differently recognized by the immune system and you can kind of avoid the immune system in that way?
Yes.
Mm-hmm. That's kind of what that first paragraph is getting at. And then I guess it's just saying that these proteins are PFE-MP1 enhances the palace serum survival by binding infected erythrocytes to end of healing. Oops. Okay.
So they're in order to improve the parasite survival, this protein binds to the infected erythrocytes that are on the endothelium. Sorry, the adhesion molecules.
Yeah. Okay, and then it says that this gene, the specific gene expressed, switches throughout an infection, and then this kind of goes into the whole like idea of how it like has different introns and exons so that it can be kind of like a different gene every time it's expressed.
Mm-hmm.
Okay, so let's go look at the traces again, because I think I probably understand why you picked C.
Yeah.
Yeah.
You switch a little more down. Yeah.
So I think what it's like focusing on is this like switching. Like, the switching of their gene expression most directly benefits them. So like, why would you want to change out the gene that's being expressed? And I think that's really what D was getting at.
Because it's just because the VAR gene is a part of the like it's helping that expression. So without the expression, it would completely take out that activity in general.
Can you say that again? Sorry.
I think it was more of not knowing what does it mean by switching of the expression. Does it mean like if it's on or off, or if it's another VAR gene?
I think it means like which like splice form. Because so you know how it was talking about like different introns and exons are getting incorporated, right? So like sometimes your VAR gene is gonna look something like this, and then sometimes your VAR gene is gonna look something more like this.
Mm-hmm.
And like one immune cell will be like primed to recognize this,
Mm-hmm.
but like um not recognize that. And so that's why you have this like switching out. And if affects, I think by switching, it just means like which form of VAR is being expressed.
Oh, okay.
Um, and so let's see what it says for C.
I just thought it would have
Yeah.
that effect with that adhesion molecule, because I thought that was the expression of VAR was with that molecule. So if it's not being
Mm-hmm.
expressed, then there's going to be, like how it said, the adhesiveness would be lower or higher because of that expression based on the VAR gene itself.
Mm-hmm.
But I
Yeah.
didn't think of it more as the overall immune system, just more of the role of the molecule itself.
Yeah. I think it was more like um maybe almost more like semantics, where I guess you were thinking of switching in terms of like on and off, and it was thinking of switching in terms of like form A versus form B,
Mm,
um, kind
mhm,
of thing. And so maybe that's why it was like slightly confusing. If you scroll just a little bit up in the paragraph, we can
mhm.
see if it mentions anything. Yeah, so I guess it's talking about the specific VAR gene expressed and consequently the antigenicity of the specific PFEMP1 on the erythrocyte surface switches throughout the infection.
Mhm.
Yeah, so I think it's really getting at that sentence right there because it mentions this idea of like antigenicity. But I can see how you like, I think you had the right thought process here. I think it was just the word switching that threw you. Um, and the way that they used it in versus the way that your brain like thought of it.
Then also going into like the transcription inactive state and the markers for the heterochromatin and then activation of the single member. So then I thought it was inactivate versus activate again.
Mm.
But I don't know, maybe that was
Well,
more
because
something else.
the transcriptionally active state. Yeah, because I guess like it doesn't say anything really about how like if the introns or the inactive parts are included, like if that affects the binding function at all, right?
I understand. Yeah. Oh, right, because
Um,
we're talking about the gene, not it's before the splicing too. Okay.
hmm. That almost makes sense. Like we don't quite even know how this kind of different expression, if it at all affects that like actual like protein function to bind erythrocytes to the endothelium.
So then, God, it's always like trying to pinpoint like picturing this without it having a picture. So the VAR gene is encoding the surface adhesion molecules, which expresses the survival of this parasite. after it's affecting the erythrocytes of the endothelium?
So I guess like this bacteria would enter into an erythrocyte. So it would be like inside like a red blood cell or something,
Right.
like causing an infection. And then it's going to do its own like transcription translation thing with
Mhm.
this VAR gene. All that splicing is happening like in here.
Mhm.
And then it's going to express some form of the
Mhm.
VAR gene out here that still has that ability to like bind to an endothelium wall and like do that function, but it's just going to be like, does it look like this or that or, you know,
It's that
this?
variety of the
Mhm.
antigenicity. Okay.
Yes, but it doesn't actually tell us anything about can this new molecule still bind to like the
Endothelium.
endothelium wall.
Okay.
If you
Okay.
just want to jump back to the answer choices, I just want to relook at the wording and see. Yeah. Okay.
I mean, that
Because
makes sense.
what I also want to point out here is it talks about like reducing the parasites. And I'm pretty sure it said that like the parasites survived by binding to the wall. So like binding was good, right?
Hmm.
So if the diversity is reducing the parasite's ability to bind, that would actually probably hurt the parasite. And we're looking here for something that's going to benefit the parasite. Yeah, because those VAR genes are leading to adhesion, and that's good.
Right.
All right.
But
So does
isn't
that make sense? Ooh,
but isn't that like it's saying most directly benefits P. falciparum by reducing the parasites? I don't know. I still don't see how it's connecting to the overall immune system versus the the adhesions of the molecule.
mhm.
Oh no, that's where it talks
So
about like like you were saying the antigenicity variation, right? Okay.
mhm,
Switching between
that's
those.
going
Got
to be like that.
yeah.
Yeah, because you're going to have like this is your immune cell, and it's going to recognize like triangle, but it's not going to recognize square. You're going to need to generate a whole new immune response, and then you switch it to circle, and you're going to need to generate a whole new immune response, that kind of thing.
Okay.
So it's almost like to the body a new infection every time because it's like, oh, I haven't seen this guy before. I've got to make like new antibodies and like start this whole process all over again.
Hmm. Understand.
Okay, cool.
Okay. So
I don't know
this
if
one,
that one almost like reading or
yeah, I think it was more of reading. This was more of being expression or switching. Okay, this one, yeah, this one was dumb. I couldn't. So it's saying what type of filament is involved in transport of our genes, hetero to the active. It was actin in the passage, and I just labeled actin wrong as an intermediate filament versus a micro.
mhm. Yeah, this is just one of those like pseudo discrete, like what is actin questions, basically.
Yep, so that's on me. Didn't know slash silly mistake. Actin. Yeah, I'll just say actin. Don't say intermediate. No, microfilament.
Mhm.
Yeah.
So it should be pretty easy to remember, like microtubules, tubulin.
Yes.
Um, I feel like that one's easy to remember. Actin, microfilament. I don't really have a not like you just got to kind of remember that one. And then the intermediate ones, I feel like are the like I don't know. To
Less.
me, they feel like the less important ones.
Um,
Um, but you're gonna think of more of like your like keratin and like the ones that you hear less about.
Gynin. No, not keratin.
Gynin.
Gynin is microtubule. My bad.
Uh,
Yeah, keratin. Okay.
yeah, cool.
34. The information passage suggests that which symptom or characteristic of malaria is PFEMP1 mediated. So again, PFEMP1, it's talking about adhesions.
Hmm.
So
So it would be kind of like which symptom is related to adhesion.
yeah,
I don't think it actually like says us any like it's all inferred, like it's anything specific.
right.
Um,
Yeah,
yeah,
I knew it
mediates the
was,
attachment.
yeah, I knew it was A or B.
Uh,
And I thought because of, I just wasn't sure because of the blood vessel blockage, whether if it's going to be like, I don't know. And I thought also like low red blood cell count, kind of if it's like there's going to be not as much adhesions if there's lower blood cells, but that doesn't matter because the adhesions is within the blood cell, so that wouldn't matter.
mhm. Does it make sense how the um the binding to the walls would block the vessels?
Because there would be like more surface area taken up, right? So less blood flow would be able to flow through because there's more resistance.
Hmm. Yeah, it's like almost if you have like plaque building up in like cardiovascular disease, same concept.
That's okay. That's what I thought, but I wasn't sure if that was actually like I thought of a arteriosclerosis basically because of
Hmm.
that plaque buildup, but I don't know why I talk or how I talked myself out of
Hmm. Yeah, I think with the like I guess reason I would go against B would just be like I don't think it actually says anything that lets us to believe that like blood cells are dying or that the like that has any effect on like actual blood cell numbers. So that's like the jump that I don't know if we can quite make based on the passage.
it. Makes sense.
Okay.
Is that more reading comprehension as well?
Um, yeah, I think just kind of thinking about how it doesn't necessarily tell us anything kind of supporting B, but there's a few things kind of hinting at A.
Because I thought it would also be like directly towards malaria, so then I have to search through and see, okay, what is malaria and how would this be affected through the adhesion process? But it's really more centered on what would happen to initiate like a disease based on these adhesion molecules.
Mhm. Yeah, it's almost asking you what does the PFEMP one actually do? So like how could that really lead to a disease?
Okay. Oh, I got this one right. Would you look at that? Oh, I remember this was very much of a guess. Losses adhesion to endothelium will most likely first flow into which of the blood vessels? I think it was something that it talked about venules, or I don't even know. Because this one, yeah, it took up a lot of time for me. So I was trying to figure out what was the process.
Yes. It says at the very last, that first paragraph, binding of infected erythrocytes in
And
brain
brain
venules.
Yeah. So we have venules. So then if it's
Cool.
venules, it's either going to be veins or capillaries. So I was like, well, capillaries doesn't make sense to me, even though I was kind of leaning towards it. But arteria is more based on the resistance. Arteries are rich in oxygen, but we were talking
Hmm.
about brain venules. So I thought it was veins or capillaries, but I'm assuming that it did end up having to go through gas exchange already. So it's already weak in oxygen.
Hmm. And if we think about the order it goes in,
That too, yeah.
it's all flowing like this way, right? Arteries into veins.
Yes.
Very um, but if you think about this, like if it's binding to the venules, venules, the capillaries come before that, right? It's like arterioles, arteries, arterioles, capillaries, venules, veins, that kind of thing. Oops. Um, so if it's flowing this way, it should like pretty much have to go to the veins. Like it can't really flow against the blood and back into the capillaries.
Yeah. I think though, again, it was the wording like likely first flow
Hmm.
because how it said in the passage was brain venules. So then I thought, okay, is it first flow before the venules? Or are you talking about after the venules?
Yeah. So I think if you think of it as like they're attaching to the venules because they're attaching from the outside, right? Oop, nope, that's not what I meant to do. They're attaching from the outside and then entering in.
Hmm.
So it would come from like X spot over here in.
I understand. Yeah, that makes sense because it would adjust or like fixate right on that. And then it's everything after that would be affected rather than
Mhm.
before.
Yeah, because it's almost like it's not really going to interact with that kind of upstream bloodstream just because it's like adhering and entering into the bloodstream like at this point. So we only really need to worry about like downstream.
Okay.
But yeah, I think that was solid, like pretty solid reasoning in picking veins.
Well, I got correct for the wrong reason, but let's, but I'll just try just as a preempt just to review, freshen up. It's never going to hurt to have one.
Mhm. Yeah. And just, I don't know. I once you kind of know that, if you just know that nothing's ever going to flow like backwards, then it's pretty straightforward from there.
Great.
I need a pair.
Oh, freaking this one.
This
Oh
is
my.
some genetics.
Yeah.
Yeah, I remember this.
Black. We were going back. All right. I think it was like 50-50, 2 out of 4 for me on this one. I don't remember, but
Mhm.
normal vision, talking about red or color deficiency, if it's sex-linked traded, affecting blue, separate but closely genes on X chromosome. Then it's talking about basically helping to read the genotype and their meaning, the plus minus, quotation mark, single quotation mark, negativity, partial versus completely, partial versus all loss. And then going into the questions, so when fewer visual pigment molecules are available to absorb light, color weakness, which of the following nervous system responses occurs?
Okay. So this question is really just asking you about signal transduction. And what it wants you to know is when you're in the immune system, when you're in like the brain
Yeah.
and like neurons, are signals like, I guess, quantitative or qualitative almost? So, like, is a signal going to have like degrees of signal? Some are weak, some are strong, or is a signal going to be kind of like on or off? And that's really what it wants you to kind of get at with neurons and action potential here. So,
Oh, that's why it's the
if you
all
look at
or nothing. So it's just gonna be you're gonna get it or you're not gonna get it.
exactly, So when you perceive something weakly, that just means it is like less of signals firing, but each of those signals still has that same like action potential curve that you know. Because, once you start that whole depolarization process, you're going to go up and hit that like peak. And it's
Yes.
going to be that same kind of peak regardless. So, what it's really getting at here is just, can you have signals that are different strengths, or are your signals more on and off and you just have a lower number of signals? So that's what like A versus B is saying.
Wow, that makes more sense than what they were saying because it makes sense if it's fewer signals then. Because the
Mm-hmm.
intensity doesn't matter. You can't, like you said, you have to reach that threshold point of when sodium channels close and when potassium starts.
Yeah. So if it ever tells you anything, because this is like saying like nervous system responses, if it ever gives you anything kind of nervous system related, jump straight to that like number, like quantitative kind of thing, rather than like how little or how much is really what that's looking for.
And- Okay.
Does that make sense?
Yeah. That,
I
yeah.
feel like that wording is also like, I feel like it's definitely very tricky, but what they're really trying to ask you is like a very simple concept. They're just trying to like disguise it as a very tricky wording kind of question.
Because the one was saying fewer visual pigment molecules, I automatically would think, okay, cones. So it depends on how many cones would be present. And if it's you're gonna make it like a weak, like if it's gonna be a strong green or more like a hint of green, for example, if that makes like the weak green, if that makes sense.
Yeah. Well, I guess if you think like this is normal and that's your cones, like you'd have like X this many signals, like all of that, this would be like 10 signals or whatever, right?
Hmm.
Versus if you have a deficiency, you're getting like 3 signals or something like that. So it would still kind of be that same like, 10 cones are all going to respond to green and fire off 10 signals. Versus if you only have 3 cones, those 3 cones will respond to green, but they're going to like not fire off, or they're only going to be able to fire off 3 signals because they're just 3 cones.
I
And then you'll
understand.
only have like 3 cones for green, recognizing that kind of thing.
And again, it's that quantity of the all or nothing because some aren't even gonna be able to show up to show that presentation.
Mm-hmm.
Under okay,
Exactly.
okay. All or nothing neurons. Um,
I don't
I
know
know
if that was more of like the tricky wording kind of thing.
I know the concept, I
So
promise. I
it
know.
might maybe just like reading just because it was kind of like how they worded it was hard.
uh I promise I know neurology, like easy peasy lemon
No,
squeeze.
I they
It's just
need they purposefully make these like if they just asked you straightforward questions, like you would be at your like target score kind of thing.
yeah.
It's half the point is interpreting the obscure language that they use.
But that's all with practice too, right? So you'll see more of
Yeah.
that pattern in question packs and section banks too, right?
Hmm.
Yeah.
And I
Okay.
think with bio too, it's helpful to be able to pick out some of those buzzwords. So like it said kind of like nervous system immediately should clue you into like, okay, I'm thinking about like um my action potential diagram and like how can I fit that kind of into the context of this question.
Okay. I understand.
Cool.
Oh, this took me 4 minutes. I don't, oh, I remember why this took me 4 minutes. This one, I know that I got this one right, but could we still go over it? Because I think I was
Hmm.
confused myself.
This is a
Um,
confusing one.
in the information below, which of the following statements best describes the color perception of a person with the phenotype red pigment absent? So I was trying my best to separate the infrared spectrum from physics and pinpoint to just on this. But it gives us that red underlies around this area. I don't know if you can see my cursor, but like around this
Yeah.
area. And then it flows into the orange at 99 of the light absorption, yellow, and so on. But then when it's saying red pigment absent, it's like,
Yeah.
does that,
So let me talk you through what I think this graph is trying to get. So it talked about how the cones, or like your cones recognize three main colors, right? So we have blue. I'm gonna call this guy blue,
Mhm,
and then green.
mhm,
This one would be green, and then
red.
red. So it should make sense how like the blue cones are probably responding to like these like low blue violet type wavelengths, and then the red cones are responding to these like
Mhm,
high red orange type wavelengths. So this is asking what happens if you don't have this like red curve. So you don't have any red pigment. You're only able to sense this blue
and the
and
green.
then this green.
Oh, that makes sense. Okay.
And yeah, this is like such a I don't like. And then you have like some kind of red stimulus. So it's
Yes.
gonna say like, what are you gonna see here? So okay, I didn't quite draw the green well enough to show that on this graph, but pretend like that's the better green because the green and the red kind of overlap a little here, right?
Mhm,
So what it's trying to like tell you is I shoot off a red wavelength and I'm looking, oh, let's see, yeah, color perception. I'm looking to see kind of what colors am I getting.
mhm,
Blue should be pretty normal, right?
mhm,
Because there's only like this tiny little overlap with
mhm,
red. So like everything that goes up until like this wavelength right here, you're going to perceive normally.
mhm,
But anything that's in that red zone, instead of getting red and green, you're only getting green.
mhm,
So here, instead of getting red and green would be normal, you're only getting green. So then we look at A. Um, red colors appear more green than normal, or green colors appear more red than normal.
I
The
understand.
A kind of makes sense. You can't detect that red anymore, but you can detect green. So instead of seeing something orange, for example, if you get a color that like falls right about, let's see, you get a color that falls like right here, instead of getting orange with like red and a tiny bit of green, you would just get a little bit of green. So anything that's like red or yellow or orange that falls on this side of the spectrum that normally has some red overlap is going to become more green. Does that make sense?
Yes.
Cool.
So if it
Yeah,
would
that one's like
say
a weird.
if it would say that green pigments absent would be more would be correct.
So potentially, so if green pigment was absent, that would be a little weird, right? Because you have only blue and only red. So these these colors, like towards the left,
Mm-hmm.
would all look a little more blue,
Mm-hmm.
versus all the colors like towards the right would all look a little more red.
Red. Okay.
So it would just kind of depend on like which side of this like little divide here that they fell on. So like yellows would look more red, but blues would look more blue, purples would look more blue, like that kind of.
But he
I think
wouldn't,
it
sorry,
well,
go
it
ahead.
actually wouldn't really affect violet because green ends here. It would
Yeah.
only really affect these ones. So it would probably be a little more red because there's very little actual overlap here. There's a lot more overlap on this red side of things.
But right where you drew that
So like,
line around 500 millimicrons, it wouldn't be violet, would it?
Um, I I think it would look like, let's see, because this would be like a mix of blue and red. I honestly don't really know because it's not like the normal like red, blue, green like
Yeah, yeah,
color way.
yeah, yeah.
Um,
Okay.
I don't know, at exactly 500. Um, because red and blue would be equal. It would probably just look like gray or something. But
Mm.
I don't actually know enough about how that signal would
Oh,
work.
you're
But
totally fine. That was an unfair question.
no, like this portion would be a little more blue, but then like the majority of this area would be like way more red. So yeah, if I had to pick, like if there was no green, I would say B. Um,
Okay.
just because that's kind of like the majority here, but it's a little more nuanced. But it should make sense at least for the red,
Yes,
just because it has so little overlap with blue.
I understand. Yeah, yeah. Okay. Okay,
Cool.
cool. Makes sense. Okay.
Yeah, it's like a really weird graph.
Correct for wrong reason. Vision spectra without red pigment. 38. Oh, another. Oh, this one also lasted forever. Oh, of course it did. I remember this. I was contemplating back and forth. This was annoying. Father with normal vision had a son who was colorblind with green pigment absent, so basically backtracking from in a Punnett square, even though Punnett squares are complicated, but colorblind with green pigment absent. So green pigment absent gives you G minus R plus.
Mhm.
That is what the son has, except the mother and father has normal vision,
Mhm.
meaning normal vision is G plus, R plus, and you have a son. So you know that genetically, the Y chromosome from the father was given to the son. So basically, at least that's what the son would have on the Y chromosome. On the X chromosome, the mom has to carry an absence of green.
Mhm.
And I don't know why it was hard for me to go between if the green pigment was absent or green pigment and the red pigment was absent. I don't know why I was debating between those two for so long.
Well, if the red pigment was absent too, because in case of A, the son would have gotten this allele, right? The G minus R minus. So he would be red deficient and green deficient in that
right.
scenario.
Yeah.
Um, because like you wouldn't be able to get like one or the other in that like scenario A. Um, like he's either getting the G plus R plus or the G minus R minus. And since he only has one of them, we know that he has to have like a G minus R plus,
Mm-hmm.
which is the only C is showing that.
And if he were to have both absent, it would be A.
If he were, yes.
Okay.
Technically, the mother could have been like heterozygous for that R minus, but it couldn't have been on that same allele as the G minus.
I
So
understand.
if C said like G plus R minus and then G minus R plus, that could make sense
Mm-hmm.
because we don't actually really care about whatever X the son didn't get. We only really care about that one with the G minus, and we know that that needs to have a like intact R because he's not red color blind.
Okay. No matter what, it would have to be the opposite because only one is present and not the other.
Mhm.
Okay. Yeah, I had to
Yeah.
backtrack and I did like a backtrack of Punnett squares. It was a mess. I know I did extra work for it, but I knew that I knew the answer. But
Mhm.
it's um, okay. The genes for red, green color blindness will affect production of proteins in which of the following cells. What was my thought process for this? Oh, That's what it was. I knew that because it was saying red color will affect production of proteins, which are the following types of cells. Well, color is for cones. And I know before looking at the choices, I thought fovea because I knew that the fovea is rich in cones, but none of the answers show fovea. So like, oh, okay, that sucks. The visual center doesn't have any, like it won't designate color blindness. Same with the optic nerve. Those are just visual centers more of, here's the picture. Optic nerve is sending signals from the eyes. So the iris versus the retina, I think this was more of, let's see if you know what the iris versus what the retina was. But the retina was really weird to me as an answer choice because of the rods. It rods is black and white.
Mm-hmm.
So that's why
Well, the
I wasn't.
retina contains the rods and the cones, though, right?
Oh, good point. And Fabia's only cones. That's true.
Yeah, because the fovea, or however you pronounce it, is like a part of the retina, right? Because it's kind of like that central point, but
Mm-hmm.
it's still kind of contained within this whole like, okay, this is the retina, like visual kind of reception site of your eye. And then here's your like specific like pinpoint of all your cones, but that's still kind of contained within this whole retina.
What's the role of the iris again? Is that just more of knowing that light goes through and is focused, or it perceives light?
Um, I mean, I think it's more of like the size of your eye,
Mhm.
um, like how much light it's letting in versus out. Yeah.
Okay, so okay.
And the pigment cells of the iris, that's just like your eye color, if that makes
Great.
sense. So
Hmm.
like I have like brown pigment cells in my iris or whatever, um, not related to how I perceive color.
Okay. Honestly, I'll tell myself this is more of a didn't know concept scenario. What was the question again? The genes will perfect. So what would be the subject? Is it more of like what is
I think
retina?
it's like retina versus iris. Yeah. Well, like what's each one doing?
Okay. All right. Passage 8, reproductive physiology. Oh, this classic one. I remember when I first took the test, this was my worst passage.
Mm-hmm. I, yeah, that's not surprising. This one's kind of terrible.
It's funky. It's weird. I didn't like, yeah, like you said, it's terrible. Um, both pineal gland secretions, high environmental temperatures, then seeing how the interactions between secretion and environmental temperatures, they went through pinealectomies versus sham surgery. And then also seeing how the pineal gland is effective of temperature when hot versus normal conditions and with or without the pineal gland in the rats or mice.
Mhm.
The rodents. Okay, going all general. Uh, table there, which of the following organelles will be relatively more abundant in the adipose tissue than in typical white adipose tissue? Well, it said in the passage, is the primary cytogenous thermogenesis heat production. Heat production means ATP. You have a lot of ATP. So I was thinking about shivering because of thermal regulation, and that requires energy. So then I led to that regarding ATP, and then that's why I led to mitochondria, because that's where ATP is made.
Yep. It was really I wanted to clue you into this idea of heat production. How like that actually works is in your electron transport chain, um, you know how you have all your complexes, you have your proton flow in and out. You're making that like proton gradient. So these are all my protons up here, beautiful. Um, those are building this gradient that you're then pushing through to make ATP. That is like basically this protein that creates a pore or like a hole in the um, electron transport chain so that protons can come through and it kind of dissipates this gradient. So you actually have to pump more protons through to refresh that gradient. And so that's actually what's generating heat, is that gradient is working harder and you're pumping more protons to make this gradient. And so that's why you are kind of generating heat throughout this process. When the protons also go back against or pass through the membrane following their gradient, that's just like being released as heat instead of being captured as ATP. So that's actually how it works. But I don't think you actually had to know that for this passage. It makes sense kind of how ATP heat is all going to relate into the mitochondria.
Mm. Cool. That's good to know, actually.
I feel like sometimes they like offhandedly mention at like the brown adipose tissue, and so that should kind of clue you into that like ATP mitochondria kind of idea.
That makes sense. And this was more abundant. Okay, so this one was right. Straightforward.
Mm-hmm.
41. Which two of the following procedures served as controls? So I knew off the bat that pinealectomy is obviously not the control.
Mm-hmm.
And same with hot versus normal temperature. Hot wasn't. So I knew 2 and 4 weren't true. And then 1 and 3 are the only ones like one of each, basically. And also this one's with the pineal gland and at normal temperature. So
Mm-hmm.
more of
Should
experimental
make sense.
analysis. Yeah.
Mm-hmm.
Is there
Easy.
42. Oh, this one took me forever. But according to passage, pineal gland has what effects on thermogenesis in wintering natural populations of golden hamsters?
Mm-hmm.
This was a lot of let's see what's comparing with what and if it makes sense.
Mm-hmm.
So that was a lot of back and
Yeah.
forth, which was annoying, but
Yeah. And if we just look at the wording of this question, we're looking at the effect of the pineal gland. That means that we have to compare some like the
normal.
conditions have to be like sham versus like the PX, right?
Mhm.
Because you can't compare the effect of the pineal gland if you're not having a condition that's like with versus without.
Right.
So you should hopefully be able to like, so okay, probably not A in that case, and then probably not D in that case. Because both of those are comparing like within two hamsters, both without the pineal gland. So then it's just looking at B versus C.
Right. And we're in wintering conditions, so technically that's cooler. So normal.
Mm-hmm. Yeah, it doesn't necessarily tell us anything about like the hot experiment will tell us something.
Um, per se. I think 43 I got wrong. Yeah. This took me forever.
Oh,
Yep, 6 minutes.
classic. Yeah, this is the classic like worst question of this practice test. Like everyone struggles with it. So don't feel alone.
Consider an experiment in
Okay.
which normal golden hamsters are injected with either pineal gland extract or physiological saline. What would not validate the results of the first experiment? So if this happened, what do you expect out of this situation in order to prove the wrong out of the results from above, basically?
Mhm. So in the above, we had this whole sham versus PX, right?
Yep.
And now we're talking about PGX versus PSAL. So in this first case up top, it's like normal versus none.
Yes.
And then in this second case, it's like extra versus normal.
Mhm.
So if we're kind of thinking about, I guess, relative levels for the sham, that's going to be like, I guess there's present pineal gland versus PDX, that's going to be like excessive. Um, and then this is non versus normal. So
Right.
it's weird about these is you have to kind of compare the two higher ones with the two lower ones.
Right.
So we're almost thinking about like we want the sham and the PGX to be similar, right? Because in both cases, those are going to be the like higher expression. So PGX and sham should be about the same. And then PX versus PSAL should kind of have the same trend as well, because we're looking at like either none versus like lower levels in normal.
Yeah.
So when we look at these trends here, we want to pick like which one matches sham and PGEX and PX and PSAL. So if we just look at like the table, we're kind of looking to you know, like look at that trend.
Mm-hmm.
Okay, let's look at which one did you end up picking? B. Yeah, I think this is just like reading the graphs from there. The tubitary weights of the PGX hot hamsters are greater than those of the PGX normal hamsters. So we want to look at like or pituitary. Kind of comparing these, right?
Mm-hmm.
So this is saying that like sham has less than PX.
Mm-hmm.
Pituitary weights of PGX hot hamsters are greater than those of PGX normal hamsters. Oops. Normal hamsters.
I hate this. This is like,
Hurts your brain.
yeah, I'm just like 60 different ways to look for these.
Oh, you know what it's saying? Never mind. Okay. We break this down. We said PGEX was like sham, right?
Mm-hmm.
So we're comparing sham hot versus, oops, it's kind of overlapping, sham normal. So sham hot versus sham normal.
Oh,
Does that
that's wrong.
make sense?
They're about the same.
Because this is comparing, I guess, within the same condition.
Yeah.
So even if you were to look at these ones over here, hot versus normal, they're still the same
They are.
because
Yeah.
your kind of relative pineal expression is the same. So you
Yeah.
have to compare within like this group versus this group, and then this group versus this group.
PGX
Okay.
P normal or
Yeah,
none.
this is like infamously a terrible question. So
But then when it's saying PGX, that's sham hot of testes, sham hot of testes, 0.8. And then PX hot, 0.8 versus 1.4 for testes. No, wait, what? No, I'm confusing myself
okay, let me see. Let me grab my, and I'm going to change my color here. Okay. So we're looking at,
now. I mean, yes, those which would not validate.
yeah,
So
I
we,
hate this question.
so we sham hot and PX hot.
Am hot and PX hot for testing. And we're looking at, we think that sham would have a greater weight. Okay, so that makes sense because that's wrong.
Right, it's the opposite. It would be true if it's less,
Mm,
not greater.
yeah. So my advice for this kind of thing in the future, this is a like cut your losses kind of question. Flag it, come back if you have time, but I think it's much more worthwhile because especially if you ran out of time in bio to get to like the last 5 questions than to get this singular one correct. Um, and this is just one that's like, you're just gonna have to sit there and like try to think about numbers and like point stuff out, and like it just gets so confusing. Definitely the case where it's probably just easier to flag it, come back if you have time, but like you're honestly better off just taking your random guess and trying to finish your section first.
Mm-hmm.
So that would potentially cut down on that, like, 6 minutes kind of thing.
Yeah, I mean, that's completely valid. Yeah.
The hard part is like recognizing that before you're like already 4 minutes in, and then you're like, oh, you know. But if you can tell that, like, okay, I'm going to have to be comparing a bunch of numbers, this is like taking a lot of brain power. Well, let me see if I can come back to it. So yeah, I guess that one's like data analysis. But
And
I will say that that's like infamously not like infamously one of the hardest questions.
there's a flag and skip if lots of analyzing of info is needed, basically.
Because like if you can just quickly glance and you can see like, okay, this one's higher than that one. But like if you're stuck there, like I'm trying to match up which one's this, and like it doesn't make sense, and like all that jazz, it just
Yeah.
gets to be a lot.
It's just not worth it then at that point because it's just going to get more complicated. And it's like, why?
Mm-hmm. Okay.
Okay, which one's this one? Oh, the individual cells. Oh, I automatically saw single-celled organisms and I thought of bacteria.
Mm-hmm.
Bacteria do not reproduce by mitosis. They use fission, go through fission. They do have subcellular organelles.
Mm-hmm. Although some like of these single-celled organisms can reproduce. It's like doesn't like sometimes it might not. It's all, and we also know that like your own cells can go by mitosis, so like that wouldn't make any sense anyways.
I mean, they would have to. Wait, isn't paramecium? Was it in this when it's saying single-celled organisms? I mean, no, I thought of bacteria.
I mean, it can be bacteria, it can be archaea, it can really be anything that like it can be bacteria or archaea basically.
Oh.
But I think A could be eliminated immediately just by knowing that because it's saying only the latter. So
Yeah, that's the part I didn't understand was only the latter. That's why.
okay, so this is saying like what can single-celled organisms do, but your tissues cannot. So this is basically saying like only single-celled organisms can dot dot dot. A, reproduce by mitosis, B, have cells, blah, 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 blah. So it's like, okay, we know our own cells can reproduce by mitosis.
Mhm.
We obviously know our own cells are going to have organelles.
Mhm.
Are our own like tissue cells, individual cells making up a tissue capable of extended independent life?
They work on their own. They do whatever they want on their own, but ours need others in order to fulfill other obligations and not just, or not obligations, just fulfill other roles.
Because we have like differentiated and like subspecialized cells and everything, so they can't survive on their own. So yeah, maybe this is just the wording, but it's basically asking like what is something kind of unique to the single-celled organisms. We know that's not mitosis, it's not organelles, and it's not metabolism. Every cell can metabolize. Only these single-celled organelles can actually like survive on their own and like live, which it makes them single-celled species.
So when it's talking about only the latter, it's referencing what was the last thing listed.
Mm-hmm. Yeah, so it's saying like if it would say like the former, it would be talking about individual cells making up tissue. Versus if it was talking about the latter, it would be it's talking about. So this is former, latter of like two things. So sometimes they'll say that.
Okay.
Yeah, they'll sometimes throw that into with like their wording and be like, the mitochondria and the Golgi can do XYZ, like what is unique to the former? And then you think, okay, what's unique to mitochondria? Or like what's unique to the latter? Like what's unique to the Golgi?
Okay. So against the wording of knowing before or after.
Mm-hmm.
So I probably thought of reproduce mitosis because of the tissue cells versus
Mm-hmm.
the paramecium. Okay, this one, I remember my thought process.
Mm-hmm.
Also metabolize glucose in the complete absence of O2, which was not produced. So in a striated muscle, metabolizes glucose in the complete absence of O2. So that means we're in anaerobic. Pyruvic acid can be made through, I mean, fermentation. No, that's not what I was talking about. Glycolysis.
Right. If you're in a muscle cell, it's basically just undergoing that like rapid glycolysis to provide you with that like boost of ATP that
Oh,
lasts like
I said
30
B.
seconds.
I said B because glucose 6 phosphate is not in the muscle, but I didn't think of it as like it's an intermediate of glycolysis.
Ah, okay. Yeah, because that's like your very first step, right?
Yeah.
Cool. Yeah, it's basically asking you like which one is like which ones are part of glycolysis. And then lactic acid is obviously that like if it
Fermentation.
can't go through the rest of aerobic respiration, it's going to go, yeah, that fermentation route.
Yes. And then pyruvic acid is going to be a product no matter what, whether anaerobic or aerobic. And then
Hmm.
acetyl-CoA and glucose 6-phosphate, I thought acetyl-CoA kind of based on, what was it, pyruvate of PDH, and then going backwards.
Hmm.
But
So this is basically, so like pyruvic acid can become lactic acid with no O2 or acetyl-CoA with O2. So we know that like in this case, like basically 100% of it is being channeled kind of this route because we don't have any O2 and it's only becoming PDH, or PDH is only being activated and transforming it into acetyl-CoA with oxygen.
then glucose 6-phosphate is just more of like, I don't, so then where does G6P come in then? Just because it's part of glycolysis, it's going to be no matter
Yeah.
what.
Yeah. So if you're going through a bunch of glycolysis, you're going through a bunch of that very first step and adding that phosphate onto glucose and then going through the rest of glycolysis. So basically, any kind of structure that's produced in that whole glycolysis chain is going to be present in a significant amount. Um, so if we think of like G3P or whatever, like any of those kind of sub structures, it's just saying is going to be really present here because you're doing a lot of glycolysis.
And glycolysis is independent of oxygen.
Yes.
Darn it. Okay. Because I've just so fixated on like with gluconeogenesis and with glycogenolysis and all that, that G6P is only in the liver. So it's like,
Mm,
and then you have to go through the Cori cycle. And then that's if you, in order to get from the lactic acid and to make it back into G6P and to use glucose, it needs to exit the muscle and go to the liver.
hmm.
So then I thought of
Yeah.
that too. So that made me confused between acetyl-CoA and G6P.
Yeah. I think it just wanted you to know that like the G6P is a part of glycolysis. So like technically you're going to produce G6P just to consume it,
Mhm.
right? Because you're immediately going to go on to that next step of like adding another phosphate,
Yes.
but or like doing your isomerization, adding another phosphate. But like it's still being, I guess, produced in a significant amount, even if you're consuming it in the very next step.
That's so dumb, but okay. Because it's
Yeah.
also a product for others, or I mean, a starting material for others, like for phosphate pentose and also for gluconeogenesis and even
Yeah.
glycogen formation. So it's like it's all like
Hmm.
an intermediate.
Well,
So that's yeah.
you could also think about like a lot of like you might be accessing your glycogen stores right now, like during this like initial phase and like trying to break that down into glucose and like grab energy from that. So like you can imagine that you're going to be like generating a lot of this glucose to immediately like push through glycolysis and get ATP from. So like it's definitely being like there and being involved.
Mhm.
It's then just being subsequently like consumed. So like if I were to measure the concentration, it probably wouldn't be that high, but it is being like produced a lot to then be consumed, which yeah, I agree with you is a little bit of like, okay, the wording there. It really just
That
wanted to hint you into like acetyl CoA needs
box.
oxygen.
It's seriously
So
all about the big idea. It's not the specifics, and I'm going way much into the specifics.
well, I think you're like on the right track with that one because like I do agree with you. Like it's good to kind of get that like, okay, G6P, let me think about gluconeogenesis, let me jump straight to these steps. But um I think that that really big idea that they just want you to know is like acetyl CoA equals aerobic. So
Mhm.
I would just like maybe like flashcard or something like let's keep that in mind, like acetyl CoA aerobic. And then if
Yeah.
it's asking you something like anaerobic versus aerobic, you really want to like hone in on that acetyl-CoA side of things. And then if you were between acetyl-CoA and something else, think about like, okay, is that an intermediate somewhere? Like, where are we going? Blah, blah, blah. Like if we were talking about ketone bodies or something, then you knew like acetyl-CoA might be more involved, blah, blah, blah. But yeah.
Yeah. Okay. Um, not to go slightly off track of the test, but I had a question regarding Corey's cycle. I know it
Mm-hmm,
never really wasn't as expressed on metabolism, but because of going from like in the muscle specifically, going to lactate, why does it
mm-hmm.
need to cross the blood? Is it just because beforehand with oh crap, I forgot what it transfers to. That what was it? Acetyl-CoA can't go from muscle to liver. I don't remember what it was that can't cross.
Yeah. Um,
Pyruvate. That
it's yeah,
or
the pyruvate, I believe, is because of like, I don't know, like if it has to do with the structure or something. But like basically if it's being transported through the blood, it needs to be in that like lactate form to
okay.
be secreted out of the cell. Um, me, I'm like, I don't know if you actually need to know like why pyruvate doesn't get like itself doesn't get.
Mhm. Because I know lactate is more like it's easier to bring that energy to the
It might
muscle.
be like a size thing, like I don't
Okay.
know. Um, like why pyruvate cells? Yeah, it could be something as like simple as like, oh, we just evolved to have like lactate transporters, so it's like just impossible for pyruvate to pass through. Like that's oftentimes the case, is just like, oh, this is just kind of how the proteins work, and so we're just gonna have the structures that work with those.
Mhm.
Um, but we know that there are definitely things that can transport lactate across that like cell membrane. So there's
Right.
some kind of like, because it does have a negative charge. So generally it wouldn't be able to cross through that cell membrane. So it has some kind of transport protein that's picking up the lactate and putting it into the blood. Um, and so we know that like that's just, how the cell works. As to why it couldn't be pyruvate versus lactate, I don't know, and I don't know if that's important.
And lactate will always be formed, but you can't use lactate in the muscle, so it has to be used in order to go back to pyruvate and then gluconeogenesis, and that's happening in the liver.
Mm-hmm.
But no matter what,
Yeah,
it has to go through pyruvate to lactate until that's anaerobic.
yeah. So basically, if you, um, I'll just draw because you kind of have an empty screen here. Um, it's like when you're at pyruvate, it can go like those two directions, kind of like we talked about, and one's like with O2 versus one's like without O2. Um, and it's basically just, is this going to be acetyl-CoA or is this going to be lactate?
Or ethanol.
Yeah. Well, ethanol not really like in our own cells, right? Because we're not capable of doing that like second fermentation step.
Right.
So if we're talking about a human and we've generated lactate, that's where we are kind of stuck. So we
Mhm.
made lactate so that we could recycle our NADH,
Right.
so that we can keep pyruvate, keep this glycolysis process going and keep regenerating pyruvate and just
Right.
keep getting those 2 ATP out of glycolysis.
Mhm.
And then we're saying, okay, we can't do anything with lactate, well,
In the
in
muscle.
our normal cells. So we got to send this off to the liver to like fully recycle it. Um, Versus like obviously acetyl-CoA, you're just going to go through with your normal oxidative respiration.
Through carbohydrate without all that metabolism or yeah, phosphorylation. Yeah.
Yeah. pH into your Krebs cycle, into your electron transport chain, and then reset. Yeah. Main purpose of lactate is to just recycle that NADH, and then kind of on a broader, like longer time scale, you're recycling the lactate itself kind of eventually. But that's kind of time limited as to how much lactate and like reusing NADH you can actually do, which is why you can only do like anaerobic exercise for like 30 seconds. And then that like kind of aerobic stuff kicks in.
But that, no, that's a stupid question. That's obvious. The NADH is recycled anyway. It's going to be automatically be used for glycolysis. Yeah, never mind.
Yeah, so the NADH is recycled on the oxidative pathway by going through the electron transport chain and giving away its electrons. And
Pumping
well,
proton.
it's like being recycled into NAD plus.
Right.
I wrote
But it's
NADH, but NAD plus, yeah.
using that electron in order to pump up the proteins and etc.
So it's actually like helping contribute to your energy count. Whereas like here, you're just kind of like using lactate to regenerate your NAD plus. So you're kind of just like losing out on that energy. Like it's not as efficient. You're still anaerobic and you're still only getting these like two oxygens or two ATPs out of glycolysis kind of thing.
Yeah. Okay.
Cool.
Any review on that?
Yeah, and then I feel like the Cori cycle itself is not too complicated. It's literally just like, can you, it like goes to liver, refreshes, that kind of thing.
Okay. Little note there. Okay. I know the law of Craig's rule. Craig's rule. I'm saying it
Is
wrong.
it like Chargoff's
Goff's
or
rule. You're right. It's not
something
Craig. I
like
don't
that?
know why I thought Craig.
Craig. I would get behind Craig's rule though.
I I don't know why, but I thought A plus G equals T. I said it in my head that A plus T equals C plus G.
Mm-hmm.
Except that's not right because it's supposed to be A plus G equals C plus T. So A and T are the same.
Yeah. The way I do this is like even without the equation, just because I find it easier to just rationalize. If I have 23% adenine, then I must have like 23% thymine, right?
Mm-hmm.
That's just how pairing's going to work. So then I'm
Mm-hmm.
just going to go over here. I'm going to say 23 plus 23, 6,
Mm-hmm.
46%. Okay. If those are 46%, then the other ones must be 54%, right? And so then I'm just going to divide that by 2 because I'm going to have an equal number of C's and G's. And I'm going to say, okay, 27.
Yeah.
So I guess it's like it is the equation, but like it's just easier for me to think of it as like, okay, I know I have to have equal amounts of A and T. And if A and T make up 46%, the rest of it must be C and G. Let me
Mm-hmm.
split that evenly, 27. So same thing of just doing it with the equation, but just kind of going through it logically.
Oh, it's
I think it's with an A, Chargaff's.
yeah. I felt dumb. I was just like, I know, but I didn't play through it. This one's a lovely. Do you know what a fatty acid is? And I actually do. Thank you very much.
Lovely. Mm-hmm.
So, okay. This one's right. Structure of FA. And passage 9. Oh my goodness.
Respiration.
Oh my god. This is like this was a physiology nightmare for me, honestly. I don't know why. I kept getting confused with it all the time. But
Mm-hmm. So we're basically just talking about the our bicarbonate buffer, right?
Yep.
Okay. Yeah. I like these passages because it's like, okay, I know what the bicarbonate buffer is. Like, I can just kind of read this passage to get a little refresher. There's equation and everything. All nice.
Yeah.
Okay.
And then we have the equation that is self-explanatory. So if we have an inhibitor, it would increase the concentration. So if we have an inhibition here, we're going to have more of the buildup. See, that's the thing also with this is like in which direction. So it's also needing to know what comes from where. Either way, if this is inhibited, something has to increase on the left side. So again, this is all through oh my God, Laplace's rule principle. I don't remember
Le Chatelier.
Thank you. I don't know why I spaced out on that.
Oh, have such weird names.
I know with the French going through. Well, either way, we know that these have to increase. So knowing that A and B are out, because then they would decrease. So then the question is, carbon dioxide or water? But carbon dioxide plays more of a major role. Water will always be there no matter what. So
yeah, well, I guess the point is like water can travel in the bloodstream on its own. Like it's just water versus like carbon dioxide is what really needs this like special buffer system and kind of special transport system. So it makes sense that like we're going to have a buildup of carbon dioxide.
now this is where it goes downhill. Would the chloride concentration be expected greater in the veins or arteries? And I could not reason my way through if it's the veins or arteries, but it went through, where is it? There was like a little excerpt about how chloride concentration results in ionic imbalance is equalized by the rapid movement of chloride concentration ions into the RBCs from the plasma. So again, we know that it goes from extracellular to intracellular space. So then I was thinking of the the electron, like not the other gradient, not the concentration, the electrogradient.
Like, yeah.
But
okay.
Again, it's like arteries or veins. So then again, that will play into, okay, the concentration of carbon dioxide and oxygen. So which one would be favored? Is it going to be favored when carbon dioxide is high or when's it low? And
so
I thought because of bicarbonate, if there's going to be more leaving the RBCs and entering the plasma. So should I dry it out? Maybe it will help if I dry it out. Like you have your extracellular barrier. Okay, let's just pretend that's right. So then, and this is your intra. So this is your, sorry, I'm doing this with my mouse, so it's really sloppy. And this is what plasma. So then if HCO3 is leaving the RBS and entering the plasma, it's going this way. And then it's the opposite for chloride. So then there's that transport or like that balance of like the ionic gradient. But then the question is, how does the carbon dioxide play a role? Because if there's going to be high bicarbonate, there's going to be lower CO2.
So you're almost there.
Damn it.
So this is totally correct in terms of the HCO3 and then the Cl balancing it out.
Yes.
The question is asking us basically, when are we going to have this bicarbonate ion present? And so if you're breathing, you're going to be producing a lot of carbon dioxide, right? So the carbon dioxide is going up. So you've just respired, you got all your fresh oxygen, and now you're getting rid of carbon dioxide. So you're at the capillary beds, right? You're exchanging out your oxygen, and your red blood cells are picking up carbon dioxide.
Right.
You are now like in this scenario, you have like 100% carbon dioxide in your red blood cells in the capillaries. That's immediately going to be turned, like be pushing with Le Chatelier's principle, this equation all the way to the right, right?
Yes.
So you're going to start producing a ton of bicarbonate ion.
Right.
So after you've picked up this CO2, it's going to be transformed into this bicarbonate ion.
Yes.
And that's when you're going to get this high concentration of bicarbonate ion. That's going to then exit the cells and do this whole exchange that you drew out over here.
Mm-hmm.
But you're getting the bicarbonate ion from that CO2 waste that you just picked up. So the CO2 is high in the capillaries. We just picked that up. Now we're transforming it into this bicarb. This is happening kind of in the veins and like venials part, because that's when we're getting this like deoxygenated blood, right?
Yes.
And so now that we're in the veins, we've produced more of this bicarb. We're going to start letting that out of the red blood cells. And that's when we're going to get this actual like exchange happening.
So then this is when we're exhaling.
Or, well, because like it's going on kind of at the same time. But yeah, it's in that deoxygenated blood that has picked up that carbon dioxide. Because like that bicarbonate needs somewhere to come from. And it's coming from all that waste carbon dioxide. So it's basically asking you, like, when are you going to be having bicarb? You're only going to have bicarb really in these veins because that's where you've picked up the CO2 and started kind of generating this whole system. If you, like in your arteries, you're going to have low CO2, so you're also going to have low bicarb. So it's just going to be like nothing really going on at baseline. But then in the veins, you're going to have high CO2 originally, which drives that equation to like lower down the CO2 to create high bicarb.
Would wouldn't carbonic acid be high in the arteries?
Carbonic acid. So not really, right? Because we don't really have kind of much of anything going on in the arteries. Arteries are mostly just going to have oxygen. So it's almost like, let me just draw this. If you have low CO2 in the arteries, you're going to have, where did that go? Okay, that didn't work. You're going to have low CO2 and low bicarb and low carbonic acid. So it's just like nothing's going on at baseline.
Oh, good point. Okay.
Right.
Yeah.
So it's like, this doesn't really matter because we just kind of have oxygen.
Yeah.
We don't really have any like CO2 involved. We don't really have any of this involved. The pH is fairly regulated. So
Great.
it's not really a concern. It's just kind of generally like, okay, this is pretty low. We don't really need this. Boom. All of a sudden we get like a ton of carbon dioxide. What do? We're going to drive this equation to the right. We're going to make bicarb. That bicarb is going to exit out of the cells and we're going to get this process happening. So this is only really relevant. This bicarbonate buffer is only really relevant in your veins, because that's where you're really going to have carbon dioxide that you need to balance out.
All right, so we're going to heavily review respiratory system, specifically buffer of HCO3. I've always been unsure about this concept all the time, but now I will. This makes more sense though. It also helps if I have the picture, but you can't have the picture.
Oops. Here's your picture.
Here's your picture.
Okay. Cool.
Oh, I have to be on mouse. All right, that was 49, right? So yeah.
Yeah, 48 was corrected.
Yeah, this was basically from what we were saying.
Mm-hmm.
So O2 prefers acidic environment, which means CO2 is going to be low, but the pressure is going to be high, and wanting to balance it out with the rest.
Yeah, so we're gonna have high CO2 that will drive more production of hydrogen.
Yep. Okay.
Yeah.
So
This is basically like when you're at the capillaries, you arrive there, your cells like, I've already respired, here's all my CO2, and it's gonna be like, here's your oxygen.
mhm. Oh, I guess that was the only question I got wrong then. I thought there was more. I guess I was just unsure then, and I wasn't so confident. But their narrow being single, I remember this was um I I remember this question specifically when I took this before, because it was between B and C for me, and B was wrong because it doesn't matter because it's always going to be in contact. Plus, it's not even like that's not where the lung, like it seriously says lung capillaries. It doesn't even have anything to do with the alveoli, or at
Mhm.
least like it there. That's how the gas exchange is happening. Is that
Yeah,
like connected, and which also follows through like that maximum exposure of the whole disc itself.
yeah. And you can just kind of think of that like, I don't know, like exchange. This equation's gotta like, if we're having this exchange like up and down, they need to like be able to interact kind of thing.
Mhm. One-on-one specifically, and like that one-to-one ratio.
Yeah, and like a lot easier for particles to pass through like a bigger space than a smaller space, just kind of in a general sense.
This was all 9. So yeah, no 10. I just don't know. Did you want to go through the whole thing? Because I know it's past 3, but I'm in no rush with anything. I don't know about you.
Um, we're good to finish up. Um,
Okay.
yeah, I don't think there's too many left that you got wrong either.
I don't think so either. I think towards the end it was fine. I think there were just some hiccups in the standalone questions.
Hmm.
Um, this is another with the KD because we talked about that last time about binding affinities. It's all different subunits. Early endosome, this is how it's like bypassing, how it's getting to the Golgi. And there was something regarding, yeah, cycle between a host membrane pore 2 cycles between Golgi apparatus and endosomes, and then reaching endoplasmic reticulum. So
Hmm.
centrioles, we're not really in, I mean, it has nothing to do with mitosis here. Plastids are not present. Nucleolus is in the nucleus. So lysosome is the only one that's in the cytoplasm.
Hmm.
So
And it like vaguely mentions that it like avoids degradation or something. Um, yeah, if it was, so I guess normally the pathway would be early endosome to late endosome to lysosome, and then that's like your trash, like your phagocytosis kind of pathway. So it's basically just saying, oh, it escapes this like degradation pathway.
yeah, and invading, reaching endoplasmic reticulum to bind. Okay.
Hmm.
53. The most likely result of various alanine to proline mutations is subunit A. Alanine, I guess this is more of what's going to happen if we go from alanine to proline, and proline is one of those weird amino acids where it doesn't really have a secondary structure because it uses its backbone of
Hmm.
the peptide bond as part of its R group. So that doesn't really have a secondary, it has that kinky feature that alanine doesn't.
Yep. So proline and then sometimes glycine can disrupt the secondary structures
With glycine's achiral.
because it's actually like so small and like flexible. So it can kind of mess up the whole like, okay, we have this whole um, you know, helix going. It has like, okay, every 3 to 4 base pairs are reacting. Glycine is just so flexible that it can like in the same way that proline is so rigid and kinky, it'll like kink it out of place. Glycine can also just kind of like
Wobble
wobble
or
out of place. Yeah.
okay. So they're literally opposite, well, not polar opposites retrospectively, but
Yeah,
opposites.
exactly. Yeah, those are the two kind of like key secondary structure amino acids.
Okay, 54. Oh, I did. Yeah, okay. Several strains are responsible for the disease, and because of the availability of antibiotics, it's not recommended as a course of treatment. Um, well, GBT is a target of several common antibiotics. I don't, that doesn't really, I don't know, that's not really the, like, it's not centered around that for course of
It's very
treatment.
unrelated.
D is not related either because we're not even talking about viruses. It's more of bacteria.
Mhm.
So it was between B and C for me, obviously even shown between like what I got for answers. I just didn't know if there would be some antibiotic resistance and also disrupting translation. So I guess it's more of knowing what that antibiotic would do to the bacteria. So that's why I wasn't so sure about this part.
Mhm.
If it
Could you
would,
scroll down just a tiny bit? I want to see
yeah,
what they say for C. Yeah, because the passage does not provide any data. Yeah, I feel like it's like because the STX is just like a product, right? It's not the actual like um bacteria themselves.
right.
So it would be hard for like the product to kind of develop antibiotic resistance and like lead to antibiotic resistance because it doesn't have like genes that can mutate or anything like that. We're more thinking about kind of the like host bacteria being affected or the host bacteria giving antibiotic resistance, that kind of thing. Does that
Not
make
following.
sense?
I'm not following.
Okay, so STX is not a bacteria, right? It's just like a product of the bacteria.
Mhm.
So STX is not going to be able to develop antibiotic resistance because antibiotic resistance happens through like think of your like natural selection, kind of genetic pressure. E. coli can mutate and kind of develop resistance, but STX itself is going to just be like a product with a fixed structure. It's not going to be able to like mutate and actually change anything. Um, and then you'd like, I don't know, is the E. coli going to take back its own STX kind of thing? Like it just doesn't necessarily make sense as to how the product could get lead to resistance. It would have to kind of originate from the E. coli original bacteria itself. Does that make sense?
Mhm.
Okay.
I'll mark these later in the sheet. I just want to go through these. Okay, so this time I got it. So this time I was saying like which one would be most effective because we know this is saying high 25. Most effective, you want the lowest, right?
Mm-hmm.
Or low value for KD, meaning high binding
Mm-hmm.
affinity.
Yep.
Okay, so that reasoning was correct. Oh, this makes me mad, but I understand why I got it wrong. Because it was saying responsible for activating subunit A. Where are we? Is cleaved into catalytic A domain. Cell surface to the position retrograde domain that functions by depurinating a specific position on the ribosome. This halts protein synthesis. So it literally says that it's information of a protein. Like it's
Mm-hmm.
part of a protein. So protease is what's going to be cleaving a protein. And for some reason, I think because of C-terminal A2 domain, I thought it was exonucleus
Hmm.
because it was the end, but that was just talking about how it's split up and it has the terminal A2 and A1 protein domains.
Hmm.
Because of that's where it was cleaved, not actually saying it's at the end of the sequence. But
Hmm.
I guess this is also true, saying that this is a polypeptide change, not
Yeah,
a last nucleotide change, hence
yeah,
nuclease rather than protease.
exactly.
So that's wording, or not wording, but knowledge of those. This is more on prokaryotic expression, knowing what happens. This makes sense to me of what happens. Again, this is more of knowing what happens with the lac operon versus what happens if you have lac I.
Hmm. Yep. This is like a classic like example of the prokaryotic like operons and stuff, transcription.
Introduction of amino acid substitution result largest decrease in entropic penalties associated with protein folding. I guess this one, I think how I approached it was knowing, are we going to a polar to non-polar? How is that going to affect hydro, like with respect to water? What's going to be wanting on the surface versus in the buried site of the protein folding, and how that's going to affect the entropy in general of the protein. And this one's
Hmm.
increasing to threonine, so non-polar to polar, surface exposed. So hydrophobic to hydrophilic. So that will be not favored.
Hmm.
Yes. Okay.
Yeah, because we're basically looking for like what's the most favored,
Mm-hmm.
or like what goes from unfavorable to more favorable B.
And we talked
Okay.
about this earlier
Hmm.
about endoderm. It's not part of the mouth, even though it is a digestive tract, but whatever, it's in the ectoderm. But inner linings of inner cavities, so that follows through stomach, bladder, and I guess the respiratory system within the
Hmm.
bronchi specifically.
Yeah, because I don't know if we talked about this, but I was thinking about like, so if it's a throat, I'm pretty sure throat comes from the ecto, no, from the endoderm, but
Mm-hmm.
like mouth, I'm pretty sure it's referencing like,
The lips, right?
yeah, or like the outer, like whatever. So yeah, silly.
Because it even says inner lining of the digestive tract, and you know that the mouth and the anus are those linings. So, but I guess, like you said, it's more of that outer per se. I don't know. So does that mean that the anus is also ecto, or is that considered endo, or is that even, I don't know, that also could be like
I'm not
me.
sure. I'm looking it up. The anal canal has an endodermal origin.
Oh, so that's endo, but mouth. Okay, it's fine.
Endodermally derived. Oh, here we go. gbs.org. Okay, so like the anal membrane technically comes from the ectoderm, so your external inner sphincter muscle. So
Okay.
like I guess the kind of externally exposed parts of both the anus and the mouth, they come from the ectoderm, which I guess makes sense to me.
Yeah,
So if
that
you
makes
think of like, I guess either end of it, like is ectoderm, and then once you get in that tract, it all becomes
endo.
endoderm from there.
Well, that would have been nice to specify, but you know what? Okay, it's fine. All right, AMC. I'll accept it. It's fine.
Mm-hmm. You let it
Okay.
pass.
Yeah, I'll let it pass for now.
Okay.
Okay, so it wasn't, I guess maybe you are right that the last chunk seemed okay.
Yeah, the last chunk definitely seemed to get a little bit better, I think. No, no. I think the passages weren't as like crazy.
Mm-hmm.
And then the standalones ended up being pretty solid.
Yeah.
Okay. Um, yeah, you can finish filling out that spreadsheet for bio.
Mhm.
I kind of was thinking, I don't know if you want to go through, there were a couple questions in Chem Phys that you maybe didn't know or had questions about, or Psych Soc if there were things that you didn't know or wanted to go over. Um, we could talk about those in another session if you wanted, or we can like do more bio stuff. This is up to you, like how you want to review it.
For the questions, like for the
Like,
Chem Phys, or
yeah, because like you marked all the ones you got wrong and stuff. Like, are you kind of able to go through and like figure those out by yourself, or is that something you'd want to like talk through together?
I think a lot of those I know. And then there were
Okay.
just like, I think it was also for some that I did mark of like questions. It's more of probably wording.
Okay.
Um, or it just wasn't clear. I mean, we kind of talked about the binding affinity more or
Mm-hmm.
less, so I could review that on my own and make sure that um I understand and I can backtrack from what we talked about before.
Mm-hmm.
This one, the one, what was it, 16, that was just bizarre because that was more of like what broke up. Like it gave us a polysaccharide and was like, how will we know the product or something of the material? I don't remember the question. And then one was
Mm-hmm.
like referring to on 50, it was passage based and was talking about like how would it affect of this binding pocket if it changed from this to this. And then it didn't really explain to me, at least because I didn't have the Jack Western, I wasn't able to just to fully locate the binding pocket of what it was talking about. Or if I did, I don't understand how the answer tied to it. Like it didn't make sense to me conceptually.
Okay. So 16, right?
16's one, yeah.
Okay. Let me just pull up. Okay. Addition of which disaccharide to a solution of AgO2 NH3 would not result in the deposition of shiny silver mirror on the walls of the reaction vessel. This is a standalone. Ooh, I don't like that.
Yeah.
Okay.
I don't know
So
the like mm,
it's basically asking us what is going to associate with Ag silver, right? Because this is AgO. What's gonna, so we think of like what's soluble, what's gonna like desolubilize it. Three of
okay.
them will, one of them won't.
Mhm.
Um, and then yeah, this says I was never a huge fan of carbohydrates in the MCAT. Okay. The solution mentioned in the question stem is silver oxide. It is used to test for reducing sugars. Okay, I feel like that might be the kind of key piece of information that you were
Yeah,
missing.
that's like flashcard, right?
Um, yeah, I don't even remember that. For a sugar to be able to reduce another compound, it should be able to be oxidized. So basically, you have to look for which of these is not a reducing sugar.
Oh, so analyze which one's sucrose and that's it. Because sucrose is the only one that's non-reducible.
Yep,
Okay,
exactly. So
fine.
I think this was just the like, that's silly. Yeah, this is like so, I don't remember this at all. The but okay, silver oxide tests for reducing sugars. That could be a flashcard. And then I guess if you're looking at the differences of all of these, cellulose, lactose, like if it's just pick one out of them, like sucrose is the only one that's different because it's the only one that has this like five-membered ring. And
Yeah,
so to me, that like immediately throws me for like,
that's
okay,
valid. Yeah.
I don't know. I feel like this just seems so like like there's just stereochem differences. Like what's the real difference between A, B, and C kind of thing. But like this one, sucrose, I'm like, okay, I would literally just pick this because it's the odd one out.
Mm-hmm. Did you
So,
learn the polysaccharides and like the linkages for each of them?
um, just like, because lactose is pretty easy.
Mm-hmm.
So I knew lactose and sucrose,
Mm-hmm.
just remembering that it has this fructose, like five-membered ring,
Mm-hmm.
but they're almost all like pretty, like, I don't think I memorized the linkages or anything.
Okay.
Anything that's, anything that's connecting like six-membered rings is always going to be this like 1,4
Okay.
because that's how like six-membered rings connect. And so if you are asked about it, sucrose is really the only one that's different because it's going to connect to this two position on the five-membered ring.
Mm-hmm. Okay.
Cool. Yeah, that's like such a left field question. What the heck?
We love it.
Yeah. Okay. Well, good to know that there's like an easy way to answer it, I guess. Okay. So then when we next meet, we can focus on doing some kind of like bio type review and trying to start nail that down.
Mm-hmm.
Okay. Cool. When would you like to meet next? I'm like literally free all week at basically any time. So,
Um, I want to say Friday,
okay.
anytime before 3 or after 4.
Okay. Do you want to shoot for Friday morning, like 9 or 10?
Yeah, we can do 10.
10. Little sleep in.
Yeah. 10 a.m. till noon.
Okay. And we'll try to go through um I'm thinking for this first one, focus really on like transcription and translation, um, and kind of nailing down a lot of the like nitty gritty, like what does what and mechanisms and that kind of thing. Um, Gene
Okay.
expression, kind of from the start to the end.
That's like all foundational too, right?
I think so.
Let's look at my handy dandy. Oh, JK,
Excellent.
1B. Yeah,
Yeah,
1. Yeah, okay.
1B, and then yeah, I don't know if that's not 1B. Yeah,
Yeah.
moved a little bit of genetics, but yeah, it'll be more thinking of like going through the names of everything and like what each little thing does and regulation of that process and maybe I'll find a passage that kind of walks through that process from like start to finish.
Mhm. I'll even do like those 30, 40 listed questions on UWorld, like we talked about on that DNA and gene expression. I think that's literally what it was.
Cool.
No,
That'll be
JK
good.
was molecular or genetics and evolution. I think it was molecular bio. That's what would be most useful to go over.
Yes.
So,
Okay, yeah, if you do those before we meet, then I can like go through those and use those as well,
yeah,
um, as like a good jumping off point.
sounds good.
Okay. Awesome. Well, then I will see you Friday.
Sounds good. We'll see you Friday. Have a good week.
Okay. Thanks, you too.
Bye.
Bye.